Office hours again today, Friday, 9 to 10 in VLSB. First announcement of something I'm sure you're all aware of. 2 weeks from today, you're going to have your first midterm. It will cover all of my lectures. And you'll be getting more information about that because you don't all come to this room. We distribute you around the campus. And it's very important you go to the right room. It's very important you know your section number. And it's very important you know your GSI number, a GSI name, as well as your student ID number. Because those things have to be put down on the answer sheet or the exam. So a lot of students don't know who their GSI name. It's your lecture GSI. It's not your lab GSI. You have two GSIs. The one we're interested in here is your lecture GSI. I will be holding a review Monday, February 13th, 7 to 9 in the evening. I'll get you in a minute. Why 7 to 9 in the evening on a Monday? Because there's a lab lecture on Monday from 5 to 7, so we can't have it earlier than that. And every other night of the week there are labs, Tuesday, Wednesday, and Thursday. You don't want to have a review Thursday and an exam on Friday. So unfortunately, it's not the most convenient time in relation to the lecture, but that's when it's going to be. Monday, February 13th, 7 to 9 a.m. in VLSB. There will be more information on that. A couple of questions. Yes? You don't have to bring your old Scantron. You bring a pencil. Okay? Yes? That's the Monday is the review session.
Is there any way that's going to be webcasted?
No, I don't think so, because they don't like to do that. It's not that we don't like to do it. They don't like to do it. They being the webcasters. Is that right? It's not webcast. They won't do it. You know, this is an opportunity, the review, for you to come and ask questions. I'm not going to lecture. I'm going to answer your questions. So if you can't make it, I'm sorry, but I will try to have, I'll get into this again in a little later next week. I'll try to have some extra office hours next week. The exam week is a complicated week for me. Enough said. Okay, I want to get on and finish the subject I was talking about on Wednesday and then move on to some new things. At the last part of the lecture on Wednesday, I was talking about enzyme regulation, and I was specifically talking about modification of enzymes that results in activation or, as you'll see in a minute, inhibition versus activation. That is, the point I was trying to make was that all enzymes are not active at all times in a cell. So I talked about proteolytic digestions of enzymes. These were proteases primarily, where you actually synthesize the enzyme in an inactive form. You cleave off proteolytically some number of amino acids. The example I gave you in the figure, 93, was some 15 amino acids are cleaved off by another protease, and that produces an active proteolytic enzyme. That's an irreversible process. Today I want to talk about protein phosphorylation, which is a very important mechanism for regulating enzyme activity. Protein phosphorylation was discovered in the mid-1980s by two biochemists up at the University of Washington in Seattle. Fisher was one of them. I don't know the other one. Okay, and what you're doing in this situation is you're taking a protein. This is a protein. And this is a serine group. And that protein is being phosphorylated. Phosphorylation, this is a term we're going to hear from here on out. Phosphorylation is the addition of phosphate to something. So I'm going to be talking a lot about phosphorylation in the next 2 weeks. And the phosphate donor is this compound ATP, which we will spend more time on in a few minutes. So it'll become clear what's going on with this compound. there's a specific enzyme, a protein kinase. A kinase is an enzyme that phosphorylates various things. Well, this protein phosphorylates a protein, which is kind of unusual, to produce a phosphorylated protein where the phosphate has been added on to the serine group. Now, I think you should understand that if I take a serine group and I put on a phosphate group, there's no charges here. This is a polar amino acid side chain, but now I've added 2 charges to that group. If that group is in or near the active site of the enzyme, that's going to affect the activity of the enzyme. So I've made the enzyme structure change. Pretty easy thing to do. The product of this reaction, ADP, has 2 phosphate groups. We'll talk about that again in a few minutes. There are, in addition, enzymes which take that phosphate group off the serine. They are phosphatases. Phosphatase, yeah, that's good. Protein phosphatases. These are enzymes that work on proteins that have phosphate groups on them. They remove the phosphate. Incidentally, this is a nomenclature that you'll see quite commonly. Instead of writing out phosphate all the time, we do P sub I, which is inorganic phosphate. And it regenerates the original form of the enzyme. Now, this reaction can activate an enzyme, then this reaction will inactivate an enzyme, or vice versa. There's no way of predicting. So protein phosphorylation can turn enzyme activities on and turn them off. In contrast to all of the other methods of regulation, pretty much all of the other methods of regulation that I talked about, this is an instantaneous process. It's turning the light on. Turn the light, put the switch up, you turn the light on, put the switch down, you turn the light off. Put a phosphate, for example, on this enzyme, maybe this is the active form, it becomes fully active. Take the phosphate off, it becomes fully inactive. A very, very powerful method for regulating the activity of enzymes. It's more complicated than that in cells because some of these enzymes can also be phosphorylated. So you're getting a very complicated series of events to regulate the activity of these enzymes that can be phosphorylated. Originally, that's all described in figure 94. And there's a legend that says what I've just said, I think. Originally, serine was the first amino acid side chain that was found to be phosphorylated, but it turns out that there are 2 others that also can be phosphorylated. There might be probably 3, but let's say 2. Phosphothreonine, another OH containing side chain, and phosphotyrosine, which also has an OH. So anything that has an OH group can get a phosphate added to it, and that is on or off. Very, very crucial. Okay, so that's sort of the end of the enzyme regulation part. What I'm going to move on to now is something related to what we'll be talking about for the next couple of weeks. Give you an introduction to ATP, this sort of magical molecule that I've alluded to already. There is, in all cells, work being done. For example, mechanical work. The muscle contraction is mechanical work. Chromosomes are pulled apart. That's mechanical work. Things like that. That's actually mechanical. In contrast to the work that I've discussed already, transport work, where molecules are transported into cells or out of cells. Active transport, as I mentioned, requires an energy source. Then there is the chemical work that goes on in the cell. Remember those schemes, those big pathway pictures. The synthesis of biological molecules requires energy, generally, and that energy, in all 3 of these cases, in all cells, is essentially this molecule ATP. ATP is termed the currency in the cell. It's what you pay to do things. When you look, the structure of ATP is given On figure 96, it's given along with its brothers, and I'll mention its brothers very soon. You know, it's not that unusual a compound. It has the same components we've seen in other systems. It has an adenine base, like you find in nucleic acids. It has a sugar, which is ribose, which you find in nucleic acids. And then it has some phosphate groups, and this is where it gets unusual. ATP is actually adenosine triphosphate, 3 phosphate groups in ATP. And here they are, 1, 2, 3. What's important for you to realize and understand is that the bonding of these 3 phosphates is different. The first phosphate, which is bound directly to the sugar group, is a phosphoester bond. I have mentioned phosphoester bonds before when I talked about phospholipids. Remember, there were phosphoester bonds and phospholipids. The remaining 2 bonds are something new that we haven't talked about. These are phosphoanhydride bonds. Anhydrides come about when 2 acids react. So here you've got a reaction of an acid with an alcohol. Here you've got a reaction of an acid with an acid, and here you have an acid and an acid. So these 2 bonds are different than this bond. Generally, what happens with ATP, and I'm not going to go into the details of the chemical differences in these bonds because I leave that for biochemistry. What happens with ATP is shown on this board when the terminal phosphate group is removed from ATP, delta G0, the free energy, the energy released when this reaction occurs, minus, is 7.3 kilocalories of energy. This is a phosphoanhydride bond that's being broken. The second phosphate, if that is removed, oh, where'd it go? Where'd it go? It's gone. It's disappeared. So taking off this phosphate, 7.3 kilocalories. Taking off this phosphate, this is also a phosphoanhydride bond, 7.3 kilocalories, and that's what's shown on this board. So what we have is ADP, adenosine diphosphate, being converted to AMP, adenosine monophosphate. Again, 7.3 kilocalories of energy are released. If we take off the third phosphate, the one in AMP, and we go to adenosine from plus phosphate, only 3.6 kilocalories of energy are released because this is a phosphoester bond. Phosphoester bonds have less energy than phosphoanhydride bonds. Here's another example. Glycerol. Remember glycerol in fats? Glycerol phosphate. Here we're simply removing the phosphate with water, hydrolyzing off the phosphate group. 2.2, minus 2.2 kilocalories of energy. This is a phosphoester bond as well. This is also shown in figure 98, the phosphoanhydrate bonds, the phosphoester bonds. Now, one thing about these numbers that I should mention is these free energy changes are measured under, quote, standard conditions. I talked about that previously. If you measure the delta G, which is the free energy change under, let's say, specified cellular conditions, the free energy of hydrolysis of the phosphate is something like minus 12 to minus 13 kilocalories in a cell, let's say. And everybody says, oh, that's good, you get more energy. More than the standard free energy. Yeah, but remember, what's very important for you to remember, if I'm synthesizing ATP, Okay, we'll just talk about ATP. If I'm synthesizing ATP, what's the standard free energy change for the synthesis of ATP from ADP and phosphate? Minus or positive? Positive, right. You require 7.3 kilocalories to make ATP. If in the cell, under cellular conditions, I release 12 to 13 kilocalories, when I hydrolyze ATP, then I'm going to need 12 or 13 kilocalories to synthesize ATP under cellular conditions. So it's a wash. What happens with ATP, and I won't draw this out because I think the figure is fine for you to see this. Figure 99 shows you that this phosphate under cellular conditions, catalyzed by enzymes, is not simply released and the energy lost. If it's released, that energy is lost. What happens is there's a phosphate transfer, like up there, for example, that's a phosphate transfer. So the 7.3 kilocalories available in this reaction, some of it is being used to put a phosphate on a serine. In this particular example, in figure 99, it shows there's an alcohol acceptor, like a sugar molecule. Sugar's got a lot of OH groups on it, and the phosphate will be transferred from ATP to some acceptor, like glucose, we'll talk about in a while, to form glucose 6-phosphate. And of course, these reactions are not 100% efficient, so there is an energy loss in all of these cases, but the energy being made available by removal of that phosphate is being trapped in metabolites, biological molecules. Another important point for you to realize is what's shown in table 100. This bond, this terminal phosphate bond, has been classically called a high energy bond. And you'll see that in various books. You'll see the statement, ATP has high energy phosphate bonds. Firstly, there's nothing magic about this particular bond. It's a phosphoanhydride bond. Phosphoanhydride bonds exist all over in nature and not in nature. In fact, if you look at a number of different biological compounds, there are compounds whose phosphate bond energy is more negative than that of ATP. Like this table shows you phospholynyl pyruvate, the free energy of hydrolysis. That means to take ATP, remove the phosphate from phospholynyl pyruvate, you would release almost 15 kilocalories. And there's a whole table of phosphate compounds where the energetics of their hydrolysis to phosphate and, let's say, phospholynyl, it's actually pyruvate in that case, would be 15 kilocalories, acetyl phosphate to acetic acid plus phosphate, minus 10.3 kilocalories, and on and on and on. There are then a number of compounds below ATP on this table in the range of 2 to 5 kilocalories where the phosphate energy is the energy of hydrolysis, and those are phosphate esters, phosphoesters. So they all have lower free energies of hydrolysis. What I want you to realize and want you to understand is what can transfer phosphate to what. There is a question on study questions number 3 that specifically deals with that. The issue here is, if I've got ATP, can I transfer a phosphate to acetyl phosphate? Or can I transfer a phosphate to glucose to make glucose 1-phosphate? And this is an energetic issue. You have to understand the energetics involved in this to be able to figure that out. It's not complicated, but you should understand that. And if you can't understand or you don't understand question 14, you better see me or see one of the GSIs. Remember, they're there for you. okay, now we are really going to change direction. So now you've got the main player. The main player is ATP. The next rest of the lecture today and the remaining lectures, which deal with respiration and photosynthesis, are to tell you about how cells make ATP. Remember, ATP is you know, it's like you have no money. If you have no money, you can't do anything. If you have ATP, you can do all of the things I mentioned. So how cells make ATP is critical to the functioning in our biological world. And I've put up here a very simple picture that I'm sure everybody has seen. It's sort of the carbon cycle in the environment. We have a chloroplast. If you ever see a picture of mine on an exam or somewhere, and it looks like that, it's a chloroplast. You haven't seen pictures of chloroplasts yet. You will in the photosynthesis lab. That's their internal membrane structure, and this is the internal membrane structure of a mitochondrion. So we have chloroplasts, which are making carbohydrates, sugars, and those sugars are used in respiration. This is glucose, for example, used by mitochondria and the remaining parts of a eukaryotic cell to make CO2 and water, and energy is released. You know that. Energy is also released in this particular case. Some energy is stored. And this number is an important number. How much energy is released when sugars are oxidized during respiration? 686. So the point, one of the points here is every time you oxidize a glucose molecule completely, you get 686 kilocalories of energy. And what you hopefully are going to do is store some of that energy in the form of ATP, which requires 7.3 kilocalories of energy. So you have the possibility, not likely, of making almost 100 molecules of ATP every time you oxidize glucose. You don't come that close to it. So this cycle, and of course during these processes there are also losses. So there's heat, and what drives the whole thing? You know what drives the whole thing. The sun. Sunlight, right? Sunlight drives the whole process. Fortunately for us, sunlight is plentiful and sunlight is cheap, so it's a good deal. What I'm going to talk about now is an introduction to photosynthesis briefly and respiration more completely. So this is respiration, the overall equation. C6H12O6 plus 6 oxygens going to 6 CO2 plus 6 H2O. Delta G0 equals minus 686. What I want to point out now is an important principle, I guess I'd say. This is an oxidation reduction reaction. What do I mean by that? I mean there are components in this that I've written on the board that are going to get oxidized. They're going to lose electrons. And there are components on the board that are going to get reduced. They're going to gain electrons. If you don't know what I'm talking about, you're in trouble from here on out. That's all I can say. So we are oxidizing sugar, glucose, to carbon dioxide in this overall equation. And we are reducing oxygen to water, because every oxidation reduction has to be accompanied by a reduction. Now, if I were to write the equation for photosynthesis, it would be the same reaction going in the opposite direction. CO2 and water being converted into oxygen and carbohydrates. But the oxidants and the reductants are different. In that case, a very unusual reaction occurs. Water is being oxidized to molecular oxygen, and CO2 is being reduced to carbohydrate. Now, when I talked about thermodynamics and I talked about this particular reaction, I said that this delta G of minus 686 gives you no information on the mechanism of the process. And I mentioned liver cells and E. coli cells and things like that. The reactions of respiration, and there are many of them, proceed in a stepwise manner. That is, it's not just taking glucose and mixing it with oxygen and boom, you get CO2 and water. There's a series of reactions that occur during respiration where oxidation reduction processes are occurring, energy is being released, and that energy is being trapped, or let's put it this way, is being used to synthesize ATP. And we're going to go through all of the details of this process extensively, let's say. You'll be sick of it. I have to introduce one of the key players in the game, and it's a compound shown on figure 101. What the compound that is one of the compounds central to this process is known as nicotinamide adenine dinucleotide. And it is shown in figure 101. Okay, complicated, it's a complicated molecule. Again, the components should look familiar. There are sugar molecules, there are phosphates in there, there are two sort of things stuck together. All that's very interesting, but it's not sort of the heart and soul of this particular compound. The thing that is the most important in this compound, which we immediately, incidentally, abbreviate as NAD, thank God. And there's a positive charge on the molecule. So we will never say nicotinamide adenine dinucleotide, because blah, 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 that's what comes out as. The ring at the top of the figure comes from niacin. Niacin is vitamin B3, I think. It's one of the vitamins. So this vitamin is modified and appears in NAD as an amide form. Let me see. I've got to erase something. I'm going crazy here. This is simple. This is trivial. Everybody knows about this. So what you have at the top of the ring is the top of the structure, sorry. Okay, and then there's an amide group on it. This is attached. And and there's a positive charge on that. That's NAD with everything else below it, okay? What happens as compounds are being oxidized during respiration is the electrons that are released during this process are transferred to this particular compound in various steps of the sequence. How that occurs is shown in the middle figure, 101B, where you see there's NAD+, this is NAD+, and NAD+ can pick up one proton and one electron. I'm sorry, two electrons and one proton. Unfortunately for us, to form this compound. I'll try not to make mistakes. Okay, that is NADH. That is the reduced form of NAD. It has picked up an electron, 2 electrons, and a proton. Why isn't it 1-to-1? It's not 1-to-1 because 1-to-1, that is, why not 2 protons and 2 electrons? Because that would be too easy for students trying to learn about this compound. Yeah. No, this is attached to the ring below. Okay? This goes down to the ribose ring. You've got a ribose ring down here with this thing sitting on the top of it, okay, in this position, okay? Okay, so coming back, it's 1 proton and 2 electrons. It's unfortunate for us that this is the way it is, but that's the way it is. It's not an important point in understanding what this does. So NAD has accepted 2 electrons and a proton and has been converted to NADH. NAD has a brother. We'll talk about its brother later. It's NADP plus, where there's a phosphate group on one of the OHs on the sugar ring, on one of the sugar rings. That's actually shown in figure 101. The second sugar ring at the bottom, it's a little blackened. That's because it becomes a phosphate group. NADP and NADPH are entirely analogous to NAD and NADH. This is the compound that's used in photosynthesis. What happens during the chemical reactions of respiration is shown in figure C, where you have the oxidation of a molecule. The proton and 2 electrons are transferred to NAD to form NADH, and then NADH is reoxidized by another molecule. So electrons are passing through this compound, going in and coming out. This compound, NAD or NADH, is not broken down. It's used over and over and over again. It's not like ATP, which is broken down. There are a group of enzymes which are known as dehydrogenases. If you see the name dehydrogenase, that should immediately pop up in your mind and say, that is an enzyme that is involved with NAD or NADP. Now I'll limit it to NAD because it's too complicated to say, but whatever I'm saying for NAD is also true for NADP. So here's an enzyme, formic dehydrogenase, and what it does is it oxidizes formic acid to CO2. When it does that, it transfers its electrons and its protons to NAD to form NADH. Then NADH can be reoxidized by some other process. Okay, so there's a case where the oxidation of a substrate is linked to the reduction of NAD. There's another enzyme which you'll see in alcoholic fermentation pathway known as alcohol dehydrogenase, where here the substrate is acetaldehyde. In this case, acetaldehyde is being reduced. It's being reduced to ethanol, our old friend ethanol. Where do the electrons and protons come from for this reduction? They come from NADH to form NAD. So these dehydrogenases can work in one direction or another direction. We're going to see a lot of dehydrogenases from here on out. And as I said, you really have to get this part clear, or whatever comes next isn't going to make any sense to you. And don't get hung up on the 2 protons, 2 electrons, and 1 proton, because I can't even remember it, right? it's not significant. Just remember that this is the oxidized, these are the oxidized forms of these molecules, and these are the reduced forms of these molecules. Oxidized forms. And reduced forms. And again, why are these molecules important? Because what we're doing in respiration is we're taking sugars and we talk about glucose. We always start with glucose, but all of these sugars are easily interchangeable. Glucose, galactose, lactose, sucrose, not a big deal. And what we're doing is we're taking molecules such as sugars, which have COH groups in them, lots of COH groups, right? And we are converting these in a series of reactions, firstly to the level of, so these are alcohols, to the level of ketones or aldehydes, then on to these compounds, which are carboxylic acids, and then these two CO2, the final product. So in each of these cases, these are oxidation steps. And I could put in here NAD is being converted to NADH, and that's exactly what's happening at each one of these various steps. Can I move this board up? I can, certainly. Now, I've said that the reactions of respiration, or the reaction of respiration, doesn't occur in one simple step. In fact, we break down respiration into 5 phases. So this is for CO2. And I'm not worried about balanced equations. This is in chemistry. So what we have in respiration, and I'm going to go through each of these in gory detail, should I say. The first series of reactions are reactions known as glycolysis. Glycolysis means the breakdown of sugars. And what we're doing here is we are converting glucose, which has 1C6, to 2 C3 products, pyruvic or pyruvate. The second phase of respiration is pyruvate oxidation, where we are converting pyruvate into a compound known as acetyl coenzyme A. And I mentioned coenzyme A when I talked about nucleotide structures, and I said the structure of coenzyme A is there. I mentioned when I said coenzymes that you're going to unfortunately encounter this name again, and here it is, coenzyme A, and we'll talk about what this is. The third phase is known either as the Krebs or citric acid cycle. And what's accomplished in the citric acid cycle is the acetyl groups are oxidized to CO2, Then there is the fourth phase, which we call oxidative phosphorylation, which has really got two parts to it. So as soon as you see phosphorylation, you should recognize that that has something to do with phosphate, and phosphate hopefully maybe being used to make ATP. Oxidative phosphorylation has part A, which is an electron transport chain. Because what's also done during the Krebs cycle is as the acetyl groups are oxidized, electrons are released. Where do the electrons go? They go to NAD, most of them. There's one other compound. They primarily go to NAD, and then in the electron transport chain, these electrons, which are in NADH now, are transferred through a series of carriers to molecular oxygen. And then there is also the process of ATP synthesis. So you can see it's a pretty hairy sequence of events. It's not simple. It's very complicated, and parts of it are rather straightforward, but parts of it are not straightforward, but you'll understand them next week. Okay, I'm going to start talking about glycolysis because I have time. I can't let you go 10 minutes early. Glycolysis, as I said, represents the split. It's called the splitting of sugar. And you can look at the pathways of glycolysis in the book, any biochemistry book, anything close to biochemistry will give you the reactions of glycolysis. There are something like 10 individual steps in glycolysis, and I don't even give you those steps, the chemistry of them, because I don't want you getting hung up on structures. Structures are not important, or if they are important, you'll understand why in some detail when I go through them. So we're talking about glycolysis. Glycolysis has, as I said, 10 steps. That means 10 enzymes involved. An important point about glycolysis is all of these enzymes are localized in the cytosol of the cell. You have to be aware of where things are going on in this process. They're going on in various different parts of the cell. You've got to know where the Krebs cycle is. You've got to know where the electron transport is. You've got to know where glycolysis is. These enzymes all occur in the cytosol. The enzymes are therefore, we say, soluble enzymes, i.e., no membranes, no intrinsic membrane proteins involved in this process. Very important, no oxygen is required for glycolysis. Even though the overall reaction of respiration requires oxygen, these first 10 steps do not require oxygen. For the reasons that I've put up here, actually, sort of this one and this one, the second and third points I've made about glycolysis, have led people to conclude that glycolysis is a fairly primitive pathway. That is, it existed in simple organisms because these simple organisms may not have had very many membranes in them, but they did not require oxygen. I think it is well accepted that the earliest forms of life on this planet did not require oxygen. And we have here a pathway that we can break, which is sugar breakdown. We can break down sugar. And most importantly, the last point is this breakdown of sugar during glycolysis results in ATP synthesis by what we call substrate level phosphorylation. I hate writing this out, but I will. And this is a very, I won't say simple way, a simpler way of making ATP in cells than what I've put up here, something known as oxidative phosphorylation. So again, this has been taken as evidence for the primitive nature of this pathway. There, I think, pretty much all organisms that we know can do glycolysis. Some of them that grow in the absence of oxygen. There are organisms, bacteria, for example, that grow in the absence of oxygen, can carry out glycolysis and can make ATP, and they can live by using the soluble enzymes and making some ATP. The amount of ATP made during glycolysis by substrate level phosphorylation is not much, but these are rather, I shouldn't say they're simple organisms. I don't know what to call them. I mean, a bacteria is pretty complicated, but it's not as complicated as us. We have all our different cells and things going on. But it's perfectly possible for organisms to exist by using glycolysis for their ATP synthesis. Now, as I said, I'm not holding you responsible for the 10 steps of glycolysis. It's way too late for me to talk about the one that I'm going to start with next time. I'll just mention there are a series of initial reactions where glucose is phosphorylated, converted from glucose to glucose 6-phosphate, and then that's converted into fructose 6-phosphate, and then again a second ATP is used. So you should be aware that in the early steps of glycolysis, ATP is actually being used, not synthesized. So you end up with a carbohydrate, which is fructose 1,6-diphosphate. It's got 2 phosphate groups on it that came from 2 ATP molecules. So, you know, when I say ATP is synthesized, it is synthesized. And next time we'll go into the couple of steps that are critical in understanding how glycolysis makes ATP.